Good. So I hope you have appropriately celebrated the crossing of the equator, the 50% of the course. And today we start the second half of the course. And we continue our lecture number 8 regarding receptors and how do we carry information from outside the cell into the inside of the cell, and how basically some biochemistry will change in the response to different signals from the outside. We have finished with the consideration of so-called GPCRs, so G-protein coupled receptors. We have considered a little bit receptor on its own, but of course, if that is a G-protein coupled receptor, it begs the question, what is the G-protein? And we have even considered a little bit something about G-proteins, and today we will continue from that point. So when unbound by ligand in the inactive conformation, receptor has no surface that is complementary to the G protein, so they are not connected. Receptor is embedded into the membrane. G protein is tethered to the membrane, so you can see that on this image. So they are freely diffusing but they are diffusing being connected to the membrane. Here is a pretty neat part. So what is diffusion? It's some sort of random walk. So they are randomly moving to the right, to the left, so whatever. I guess you know what is diffusion. The interesting thing is that we have so-called Paulias theorem. That's a pure mathematical theorem, but it tells us that there is a drastic difference between diffusion in 1D, 2D, and 3D. In 1 and 2 dimensions, if we have diffusion of, let's say, 2 different particles, there is enormous probability that they will eventually meet. Well, I can put that more mathematically rigorous, so I would say that with the probability 1 there will be some moment t that they will meet. The same thing applies to the 2D, to the two-dimensional space. Very similar argument works if you say that the particle will always return to the point where it started. But if we get into 3D, the theorem says that the particle will never return to the point where it started. So you cannot tell that 2 different particles will eventually meet. So being tethered to the membrane has to do with that. And in some cases, we don't even need that membrane for something to function, but if we need different proteins to meet and somehow interact, and we want that probability to be higher, one of the possibilities is to tether them to the membrane, and that's why and make them diffuse just in 2-dimensional space, not in the 3-dimensional space. So that's a neat part. So now GPCR binds to the G protein, induces a change in the conformation. So G protein is a trimer. It consists from G alpha, G beta, and G gamma. And there is on G alpha subunit, there is a binding site for the GDP. And when it binds to the G protein, this GDP is released, and subsequently it can be replaced with GTP, so with guanine 3 phosphate, that rapidly binds into the empty, into that pocket, because we have much higher concentration of GTP inside of the cell compared to the GDP. And here I have a tricky question for you. How do you think? Why do we activate alpha subunit by exchanging GDP for GTP? That looks like a little bit tedious. So you need to release GDP, and you should wait until you get some fresh subunit, fresh molecule of GTP to bind and to make that alpha subunit activated and then do whatever I will show you. If we could instead have GTP bound to the alpha subunit, and for activation, for example, immediately release one of the phosphate and transform that just here in place into GDP. So why this pathway? Why not another way? How do you think? Make a guess. Well, there's actually nothing to do. There are no processes that we need so much energy, so it's not energetical. It has to do with the fact that one process you should wait for that exchange, the other process you can do here just in place. Let me give you a hint. If you started some signaling pathway, so some signal comes, you started a pathway, will you need to turn that pathway off? Well, probably yes, because you don't want that to be permanently activated. That would be no good. So you need to turn on, and you need to have some switch to turn off. Can you reason now in this context, why do we go this way? No? Look. Transforming GDP into GTP, that's really some sort of energy consuming process, and it's a little bit hard to do that. But transforming GTP into GDP is easy to do. So you just remove one of the phosphate and you can do that. That can eventually just happen, so it's not a big problem. So think about it. If you have activated G alpha, you have put in there GTP. You may expect that at some point eventually that GTP will be transformed into GDP and your G protein will turn off. So that's the main reason. So you are setting like some sort of timer into that G alpha subunit, and you know that after some time passes, it will eventually turn off. And that's really good, because you don't want your pathway to be indefinitely activated. So you activated that, you passed some sort of information, so you have turned it on some biochemical pathways, and then you should shut down everything. So maybe the signal terminated from the outside, maybe something other happened, but you should somehow terminate everything, and this is a good way to do that. Okay, so here is an example of that alpha subunit. Here you see GTP inside, so here you see that. When it GTP, it changes the form of the G alpha subunit. So these heterotrimeric G proteins, they directly bind to and are activated by GPCRs that function as guanine nucleotide exchange factors. So you will see that in literature, GEFs. They trigger release of GDP, and G protein and then spontaneously binds GTP, favored by high intracellular concentration of GTP relative to that of GDP. That induces conformational change involving two highly conserved segments. So here they are, segment 1, segment 2, here they are called switch 1 and switch 2, that allow the protein to bind and to activate downstream signaling proteins. But eventually this GTP will transform into GDP. So you see this bond, it will break, it will release inorganic phosphate, so this part, and you will get protein bound to GDP. Conformation of switch 1 and switch 2 will change because this phosphate goes away, so they change their conformation back, and you get your G protein in the off state. So just after a brief time, the active G protein is shut off, so converting back to the inactive form, because it has intrinsic GTPase activity, and this activity allows it to slowly hydrolyze the bond between GDP and phosphate, so transform GTP into GDP. That is performed seconds to minutes, and even this time is too large, so we will meet some more factors that make this process even even quicker. The rate of GTP hydrolysis determines the length of time the GTP binding protein remains in the active conformation and is able to signal downstream target proteins. And monomeric, often called low molecular weight G proteins, including RAS and various RAS-like proteins such as RAN and SER, do not directly bind to receptors but act as intermediate proteins in signal transduction pathways. So monomeric G proteins play crucial roles in many pathways that regulate cell division and cell motility. We will meet that. We'll even meet them, I guess, when we will consider some vision and how do we see the light. There are G protein coupled receptors involved as well. Here is an example of the life cycle of the G protein. Here you see it has some sort of GTPase activity, and it is in state on when it is bound to GTP. When this activity transforms GTP into GDP, you get state off. So you have some sort of timer ticking inside that G protein, ready to disable the signal and thus stop transducing that signal downstream to the subsequent proteins. And that's really good because you don't want your cells to be permanently hyperactivated and performing something you don't want to perform. So you need the cell needs to respond adequately. So if you have some signal from the outside, you respond. If you have no signal, you should not respond. Otherwise, it's waste of energy and different resources. The rate of GTP hydrolysis is often modulated by other proteins. For instance, both GTPase activating proteins, GAPs, and regulators of G protein signaling, RGS, accelerate GTP hydrolysis. Many regulators of G protein activity are themselves controlled by extracellular signals. So you may have one signal that activates G proteins and goes somewhere, so they activate something other, I don't know, some adenylate cyclase or something like that, and you have a different signal that modulates this one and basically tells the cell if it should respond vigorously to the signal from the outside, or it should somehow diminish its value. So not so actively respond to that signal. And that is performed by basically modulating, well, one of the ways to do that is to modulate the time that G proteins are active and are able to activate something downstream. Of course, that can be used by different bad organisms. For example, Vibrio cholera. It synthesizes certain toxins that penetrate the plasma membrane of cells, such as those that line the small intestine, and catalyzes a chemical modification of G-alpha, preventing hydrolysis of GTP. So you see what happened. It is not disabling something. Instead, it removes the GTPase activity from the G protein. So now you get G proteins that are activated and never deactivated. So you cannot turn them off. And that constant hyperactivation leads to loss of electrolytes and water from these intestinal cells into the intestinal lumen. And, well, you get pretty nasty disease. So cholera is really not good. So you see what happens. We need that deactivation, and it's really crucial to be able to stop that transduction of the signal somehow, and that timings, they really matter. Good. So different G proteins are activated by different GPCRs, and in turn they regulate different effector proteins. So all effector proteins in GPCR signal transduction pathways are either membrane-bound ion channels or membrane-bound enzymes that catalyze the formation of one or more of the second messengers. Once again, I think we now understand a little bit why membrane-bound. So we bind G protein to the membrane and we bind receptors to the membrane, so they are diffusing in 2D, and we have much higher probability that they will eventually meet and somehow interact. For example, if the receptor is active because it is bound to the ligand, so it's in activated state, it will activate G protein, and now G protein will go in the activated state and activate something else, some adenylate cyclase or whatever. So humans have 21 different alpha subunits, 6 beta subunits, and 12 gamma subunits. The 21 alpha subunits are encoded by 16 genes, several of which undergo an alternative splicing. Do you know what is alternative splicing? Did you learn that during, I don't know, molecular biology course?
Yes, we did.
Good. And so far as is known, the different beta and gamma subunits are interchangeable. So everything is more about alpha subunits. And GPCRs represent the largest protein family in the human genome, so approximately 800 genes. So you see that these G proteins are really interesting from the scientific point of view, because many genes correspond to that GPCRs, and many processes are controlled with GPCRs. And 400 GPCRs genes are thought to encode sensory receptors. Of these, the majority are in the olfactory systems and bind odorants. I guess we will eventually have a lecture on odorants, and maybe I'll tell you a little bit more about that. Here are examples of classes of of G alpha subunits and effectors that they are associated with. So what do they basically activate? So here you see the adenylate cyclase. This is an enzyme that creates CAMP, that's cyclic AMP. I guess I have some formula a little bit later in our slides. And here are examples of different receptors. So beta adrenergic receptor, or receptors for glucagon or serotonin or vasopressin, so whatever. Here we have different types of these G alphas. Probably will be mostly interested in how do they interact with adenylyl cyclase, that's one, and with phospholipase C. So there's another pathway. Here we create inositol 3-phosphate and diacylglycerol. So here they are. I guess I will show you the formulae. And examples are alpha-1 adrenergic receptor or acetylcholine receptor in endothelial cells. So I guess we will get to that. Good. Here are examples of so-called second messengers. So here they are. So these are the formulae for 4 common compounds that often function as intracellular second messengers, and a little bit more extended table. Here you see cyclic AMP. Cyclic refers to this cycle, so you see that's cyclic AMP. Here you see cyclic GMP. Once again, here is the cycle. And diacyl glycerol, here it is. Here you see how that looks like. So you have glycerol and you have fatty acyl groups, 2 of them connected. So here it is. And here is inositol 1,4,5,3-phosphate. So here is basically these rings, a ring with OHs, that's inositol, that's an alcohol, and here are phosphate groups connected to that ring, so that's why triphosphate, because we have 3 phosphates connected to there. Here you can look after the lecture, you can see the table and get some more examples of what's happening right there. Here is a nice picture when we see how adenylyl cyclase activity is modulated by the interplay of stimulator GS and inhibitor G proteins. So when we have G protein activated, that activated G protein, it can either activate or it can inhibit something inside the cell. So in this case, it activates and inhibits the adenylyl cyclase that will synthesize from ATP cyclic AMP. So it will remove 2 of the phosphates and create that sort of cycle I showed you on the previous slide. And here you see how that is regulated by G proteins. So receptor is activated. This trimeric protein, it dissociates, so we get gamma and beta subunits separately from the alpha subunit. Alpha subunit may diffuse. It will eventually meet adenylyl cyclase, and this one will activate it, and this one will inhibit. So the overall response you get will basically depend on the interplay. So how many of you have of these, and how many do you have of these? So binding of hormones to beta 1 and beta 2 adrenergic receptors activates adenylyl cyclase via GS, whereas hormone binding to alpha 2 adrenergic receptors lead to the inhibition of adenylyl cyclase. Inhibition may occur by direct inhibition of cyclase activity by GIA or by binding to GIBG or GSA. So depending on the strength of these 2 signals, you will get some overall response. Good. Here is one more picture that basically depicts the same thing. So here we have some stimulatory hormone like epinephrine or glucagon, and we have receptor. It once again activates some G proteins, so here we have G alpha subunit, and that interacts with adenylyl cyclase. And here we have inhibitory hormone like adenosine or PGA1, and we have G alpha I subunit that inhibits adenylyl cyclase. Here you see the chemical formulae once again. So what does adenylyl cyclase do? So it takes ATP, removes 2 of the phosphates, and creates these sort of cycles. So maybe it's not well drawn here, but here is a bond between phosphate and this oxygen. Good. So that regulation occurs in many cell types and provides a fine-tuned control of the CAMP level and thus of the downstream cellular response. So you have 2 different signals integrated into one downstream signal. And virtually all of the diverse effects of CAMP are mediated through its activation of the protein kinase A. This protein kinase phosphorylates multiple intracellular target proteins expressed in different cell types. So I guess I will have some examples. So here is a protein kinase A that is a tetramer consisting of two regulatory subunits and two catalytic subunits. Each of the two R subunits is an R2 dimer and has two CAMP binding domains, so it senses the level of the CAMP. These domains are called CNBA and CNBB, as well as a linker domain of unknown structure. So here is the linker domain. I guess it should have some sort of unordered structure. Uh, the most information we have about proteins, we have basically about so-called globular proteins. That's because they are more easy to crystallize and to get some sort of X-ray of that proteins. So probably our knowledge about the form and the structure of the proteins is in some sense is biased by the globular proteins. But it turns out that there are many proteins that have some unordered or some unstructured regions. So they have some sort of internal unstructured regions, and these proteins are pretty complicated to crystallize if possible, and our data on their structure is much more scarce. And that may be interesting for you even as bioinformaticians, because if you will ever work with some software that predicts the structure of the proteins, it will be probably some gigantic neural network, I don't know, some AlphaFold or whatever there is, so modern some modern tools. And the thing is that it's quite probable that it is trained on the data set that contains mostly globular proteins. So if it will meet some protein with that sort of internally unstructured region, I really doubt if it will be able to correctly predict how that looks like. So maybe at some point we will, I don't know, maybe you will solve that problem in the future. I don't know. Maybe the problem will be solved on its own if we get enough data. I can't be sure, but I'm pretty sure that there is such a bias in all the data sets, and maybe these effects are observable if we will ask these tools to predict the structure of some proteins with these sort of internal regions. Here is a small linker region, but you get the idea. We may have protein with much larger unstructured regions. Good. So binding CAMP to the CNBA domain causes a change in its conformation. Well, of course, that's a standard phrase, so something binds to the protein changes conformation. Well, if it senses the presence of that chemical. So that it can no longer bind and inhibit the catalytic subunit that releases the C subunit, the catalytic subunit, and instantly activates the kinase activity. Good. So here you see these example. So how that looks like. You have inactive protein. You have binding for CAMP. And here you have activated C subunit, so activated catalytic subunit. And here is example of some closer look, how CAMP is bound inside and changes the conformation of the protein. Good. Here is an example of regulation of glycogen metabolism by CAMP and protein kinase A. So here you see these formulae, how glycogen is synthesized. So you have UDP glucose, so-called activated glucose. That's a glucose with attached phosphouridine. So here it is. and you have some already synthesized glycogen, and you attach the thing to the growing chain of the glycogen. Well, if you know something about starch, you should know that glycogen is often called like something like animal starch, so they are pretty similar in function and in chemistry. There are certain subtle differences regarding branching, but let's disregard that for a moment. So here it is, and here on the right you see basically the regulation, how that happens in terms of different pathways. So if a stimulatory hormone, say epinephrine or glucagon, come, they activate GPCR. So GPCR activates G protein, GS alpha subunit activates adenylate cyclase. Adenylate cyclase creates the second messenger CAMP, cyclic AMP. It activates protein kinase A, PKA, and this protein kinase A activates different proteins. So, for example, glycogen phosphorylase kinase (GPK). And it inhibits glycogen synthase, and it activates inhibitor of phosphoprotein phosphatase IP. So here you see what happens. So basically this hormone tells you to do what? Create glycogen or destroy it. Look at the image. So everything is in here. So what happens to the glycogen? I see an answer. No, we do not create it. Look, we had glycogen with n+1 subunits, and now we have activated glycogen phosphorylase kinase that activates glycogen phosphorylase, that basically takes these n+1 subunits and transform that into n subunits plus glucose 1-phosphate that is then is used to create glucose 6-phosphate that is basically in the liver is transformed into the glucose. So you remember we had a similar pathway when we considered gluconeogenesis, and I told you that there are certain situations when a liver is releasing some glucose into the bloodstream because we need that for energy. So we need an energy right now immediately, and that is performed by releasing the glucose into the bloodstream. Here is one more mechanism for you. So if we have some storage, so we have this energy stored in form of glycogen, and we can do that by basically this glycogen phosphorylase. We can take away one of the subunits of the glycogen and transform that into glucose 6-phosphate and then into the glucose and release it into the bloodstream. Similar processes we may have, so there are some pretty small stashes of the glycogen in muscle cells, but there are some. And here is another pathway. If we remove that stimulatory hormone, the pathway turns off, and we have once more generation of the glycogen favored to the destruction of the glycogen. So here we have more of it generated because glycogen synthase is now released from the inhibition, and the other enzymes that were basically destroying glycogen are now turned off. And here you see that we have a situation when glycogen is created when we have no more stimulatory hormone. So when this is removed, everything shuts off. So that's what we have. Okay. Uh, Here we see the reason for having all that second messengers and so on. In the previous lecture, as I remember, someone correctly answered this question and told me that amplification is the correct word. So why do we need all that? And here you see how that amplification may what do we basically see? So we have some epinephrine, concentration pretty low, 10 to the minus 10 of moles per liter, so pretty low. But we get to the adenylyl cyclase, we can activate few of them, they generate even more CAMP that can get to the 10 to the minus 6 mole per liter. So please note, just in 2 steps we were able to get from 10 to the minus 10 to 10 to the minus 6. So that's 10,000 times more than, so 10 to the minus 6 is 10,000 times more than 10 to the minus 10. So that's a really large number, and we can have even more and more amplification. So yes, good. Here is an example of activation of gene transcription by that sort of signals. So here is an image that shows you how that happens. So once again, we have exterior, we have GS protein coupled receptor, and we have adenylyl cyclase that generates CAMP. CAMP may activate protein kinase A. So we have considered that protein kinase A. So I hope you recall this shape from the previous slides. These protein kinase A may phosphorylate some different proteins. For example, they can phosphorylate serine 133 on a transcription factor called Cre binding protein, or CRAB. Cre, that's AMP response element, which contains a highly conservative nucleotide sequence that you see here on this slide. Phosphorylated CRAB binds to CRAB-containing target genes and also binds to the activator called CBP/P300. So this CBP links CRAB to RNA polymerase II and other gene regulatory proteins, thereby stimulating gene transcription. So here you see how that works. How CRAB is activated, so that is performed in the nucleus. Activated CRAB, it basically here you see CBP P300, and everything that links RNA polymerase to that CRAB binding site, and that activates the transcription. Good. Yes, a tricky question for you. I guess you should already pass that. RNA polymerase 2. What are the other RNA polymerases you know? What do they do? Mm-hmm. 1 and 3. Yes. Yes, good. Good. Okay, so what else? Here is one more interesting example. When we want localized effects of the CAMP to specific regions of the cell. That may be yes, correct. Good. So I see you really study hard the molecular biology. That's really great. I guess we will have a lecture about that the next lecture. and we will meet there all these polymerases. Good. So here is just an example. What do we do if we want to localize? You have midterm exam. Yes. That may be complicated, I guess. Good. I wish you luck. I wish you luck. I hope you will all pass and everything will be great. Good. So you might already have some question, like if all the processes I have showed to you are leading to generation of cAMP, to cyclic AMP, how do the cell knows what to do next if this is the same cAMP everywhere? And one of the possible answer is to anchor some proteins and localize effects of cAMP to specific region. So you have a specific region of the cell, you have there localized some exact proteins that should in certain way respond to that cAMP, and you basically get that like distinguishing what this cAMP means, so what do we want the cell to do in response to appropriate receptors by localizing spatially different proteins that should respond to that second messengers. Here are examples of multiple feedback mechanisms that suppress signaling by GPCR. So I'm not sure if I get into that. So G proteins, I showed you, they self-turn off. You know that PDE hydrolyzes cAMP, so that synthesized cAMP will be destroyed by phosphodiesterase, so it's not forever. pKa phosphorylates several serine and threonine residues, typically in the third cytoplasmic loop of the C-terminal of receptors, so it cannot more bind GS, so at some point receptor is turned off, and other similar receptors may be phosphorylated, so you may have so-called heterologous desensitization. So it's when different receptors are desensitized in response to activation of one of them. And of course there also exists homologous desensitization. A complicated word. Good. There's another pathway through the inositol 3-phosphate. That's another signal transduction pathway that leads to increase of the calcium. So here you see endoplasmatic reticulum. Some of the ion channels are opened. These are calcium ion channels. So they release calcium. So here you see what happens. So receptor is activated. Once again, we have G protein. G protein activates this time phospholipase C beta. Phospholipase C beta generates inositol 3-phosphate. So here it is, and diacylglycerol. So diacylglycerol can activate a protein kinase B, and inositol 3-phosphate may activate these ion channels that release calcium, and this calcium basically may activate something more. It can also activate this protein kinase C, but there are other proteins that are activated as well. So here is calmodulin. You will often meet that, so I guess just some sort of example. It is widely distributed in the cytosolic protein, and here it's 3D structure. Okay. I guess we are scarce on time, so I'll try to get a little bit quicker. One more large class of receptors, receptor tyrosine kinases. These are receptors that have some sort of kinase activity. The extracellular signaling molecules that activate receptor tyrosine kinases constitute a large class of soluble and membrane-bound protein hormones, including that were initially identified because they stimulate the growth of specific types of cells, so hence the name of growth factor. So if you hear growth factor, it's probably something that influences the RTK, so receptor tyrosine kinase. So some examples: nerve growth factor, fibroblast growth factor, and so on. Insulin as well regulates expression of multiple genes that control sugar and lipid metabolism in liver, muscle, and adipose cells. So you will see these sort of receptors in many cases. They look pretty different, as you see. So here you see these RTK. When ligand binds, they dimerize. So here is a step of dimerization. We didn't have that with GPCRs or with ion channels, but here we have it. They phosphorylate each other. So here we have some sort of phosphorylation, and this phosphorylation, it activates the receptor. So most RTKs are monomeric, and ligand binding to the extracellular domain induces two monomers to join together as a dimer. The cytosolic kinase domain of an RTK contains a flexible domain termed activation loop. In the resting state, the activation loop is not phosphorylated and assumes a conformation that blocks kinase activity, so it is inactive. For most, but not all RTKs, the weakly active kinase in each subunit is thought to phosphorylate a key tyrosine residue in the activation loop of the other subunit. So they phosphorylate each other, crisscross, and the strongly active kinase then phosphorylate additional tyrosine residues in cytosolic domain of the receptor. Here is insulin receptor. It differs a little bit because it is pre-dimerized. So most RTKs exist as monomers, but during all that sort of diffusion movement, they meet and they dimerize. Insulin receptor has these sort of disulfide bonds, so it is pre-dimerized, but regarding the rest of the mechanism works pretty similar. I just should tell you about that receptor because it's really often used in all of the different books on biochemistry, so you will probably meet that. So here is one more nice picture of how that looks like. So here you have N lobe, activation loop, and C lobe, and here you see what happens. So they dimerize and they phosphorylate each other. Here is an example of the pathway. This is Res-MAP kinase pathway. Here you have FGF ligand dimer. So once again, GF growth factor. So they phosphorylate each other. Here you see some adapter proteins, some gaps, and that influences the pathway in the cell. So here you see this pathway shown on the right. So ligand, RTK, GF, GF to RES, RES to REF, REF to MEC, MEC to ARC, ARC to transcription factors, and transcription factors activate transcription. So here is some closer look of how that happens. So here is active dimeric MAP kinase, and that activates P90RSK. That P90RSK activates some more different proteins, so on and so on. But at the end of the day, they promote transcription inside the nucleus, and you get more RNA for something, and basically you are changing the balance of the proteins inside the cell by promoting or inhibiting transcription of certain genes. Here is one more example. How can we localize that spatially to get certain responses, to get, let's say, more actively response, and to somehow differentiate between different signals. Here you see scaffold proteins, and in this example you see two different cases. Like you have an osmoregulatory pathway and you have mating pathway, and in here and here you have different receptors but it goes it all goes to activation of MAC, then another MAC, then MAP, and so on. It goes to transcription factors, but at the end of the day we want to activate different transcription factors because we have like different signals, and these scaffold proteins, they prevent STE11 from phosphorylating the wrong MAC kinase. The scaffold protein STE5 employed in mating pathway has no obvious catalytic function but stabilizes that large complex, and and the MAP kinase in the and everything that is downstream. So here you see these two examples. You can create this sort of signal transduction pathway by localizing all your proteins in this pathway with the help of scaffolding proteins like these STE5 and that basically helps you, let's say, to differentiate between different signals, because you have some proteins are the same in the both pathways and you don't want them to cross interact and to activate, let's say, mating pathway when we need to regulate some osmotic things. So that's one of the way to get away in that situation. So here is a little bit more about phosphoinositide signal transduction pathways. So here you see that RTK activation leads to autophosphorylation of tyrosine residues, and everything I showed you, the transformation of phosphoinositol 2-phosphate into phosphoinositol 3-phosphate and diacylglycerol that diffuses, releases calcium into the cytosol. The diacylglycerol remains in the membrane together with calcium plus activates protein kinase C, and so on. So that's regarding that picture that I showed you previously that we had. Good. What else? This is once more this pathway shown to you, so I'm not getting into deep into details here. One more pathway, so JAK-STAT signal transduction pathway. That's rapid and direct signaling, so no second messengers. Typical in immune and hematopoietic signaling, and persistent activation is linked to many inflammatory and oncogenic pathways. So I'm not getting into many details here, so you see how that looks like, and the ligands are often cytokines or some growth hormones. Once again, you get receptor dimerization, and it's called JAK because Janus kinases that are bound to receptor tails and cross-phosphorylate each other. And here you see this pathway description of this pathway. So here you see activation of the STAT proteins. So here you see the dimerization was performed. Here are JAKs, so Janus kinases. Here you see STAT proteins, and these STAT proteins, they influence, they bind to the DNA, and basically they can activate or inhibit some of the genes. Here are some more mechanisms. I think I'll skip that. Let's dash through some other pathways really quickly. So I have shown to you the most prominent pathways. So we have seen signaling with GPCRs, with RTKs, with ion channels. Well, we will get into ion channels in the next lectures in more details, and now I will just quickly dash through some other pathways just for you to know that they exist and how do they look like. Without too many details, I don't think it's really necessary. So here is TGF beta SMAD signal transduction pathway. So here you see the image. how that signal gets from the outside to the inside. So ligand binds to the type 2 receptor, threonine kinase, receptor phosphorylates and activates type 1 receptor, type 1 receptor phosphorylates SMADs. So everyone phosphorylates everyone, you might guess that. So all the pathways, they look this way. A phosphorylates B, B phosphorylates C, and so on, so on, so on. So they are phosphorylating each other and everyone is doing something. So our SMADs form a complex with SMAD4. SMAD complex translocates to the nucleus. In nucleus, SMADs bind DNA, and transcriptional, co-activational, or co-repressional factors, and that regulates cell growth or differentiation or ECM production. So whatever. So here you see all that system. So how do they bind each other and activate the transcription, for example. Here are some more nice pictures of that, so I'm not going into the details. Notch delta signaling pathway. So here you see two cells being in a contact. It's a contact depending signaling, so both notch receptors and delta or JAK ligand are membrane proteins, and signaling occurs only between adjacent cells. That's a difference with our previous methods of signaling we have considered. So ligand binding and cleavage: when delta binds notch, the receptor's extracellular domain is cleaved by ADAM protease, then further cut by gamma secretase, and then NYCD fragment enters the nucleus, and NICD binds the CSL transcription factor, converting it from a repressor to an activator, and that induces certain genes controlling cell proliferation, differentiation, or even apoptosis. So here you see how that all looks like. So all that binding, nicking, and so on and so forth. And here you see how these receptors they may be degraded, and there's the whole life cycle of what happens. And this is interesting because that determines cell fate decision. For example, lateral inhibition in neurogenesis or involved in stem cells maintenance and tissue patterning, and dysregulations are linked to cancer and developmental disorders. So you might guess, if cells are neighboring and one cell is becoming something, I don't know, cell of skin or of a blood vessel or whatever, it somehow tells about that to neighboring cells, and it may induce some behavior in them. So this is one of the best ways that cells transduce this sort of information and influence one another during the development of the organism. And you know that development should be well concerted so that you get organism and not some random bunch of cells. So everything should follow some general plan. Here is example of VNT beta catenin signaling pathway. So one more pathway where everyone phosphorylates everyone, but here is a nice interesting catch in this pathway. So VNT proteins, secreted glycoproteins, bind to freezed receptors and co-receptors on the cell surface. Without VNT, beta-catenin is continuously degraded by the destruction complex. So it's pretty interesting. So the cell continuously synthesizes beta-catenin, and it is continuously degraded. So the signal does not go further because you destroy basically beta-catenin. When VNT is present, the binding of VNT to FD-LRP inactivates the destruction complex. So you basically have something that creates signal, and you have a complex that continuously destroys that signal. Then you shut down the system that destroys the signal, and your signal is able to go further. So this beta-catenin binds to this TCF, LEF transcription factors, converting them from repressors to activators of gene transcription, and that induces genes controlling cell proliferation, differentiation, and so on. So that's essential for embryonic patterning and stem cell renewal. And if this pathway is dysregulated, it's often seen in cancer like colon carcinoma or fibrosis or developmental defects. So here you see that. So one more interesting idea for you, that it may be that you are constantly creating some signaling molecule, you are destroying that molecule, and basically your signal from the outside of the cell tells you to shut down that complex that destroys that molecule, so that molecule may go further and do something useful, let's say activate some genes. Hedgehog pathway, one more pathway critical for embryonic patterning, neural tube and limb development. If something goes wrong, you get very, very bad consequences. Believe me, don't Google that. And aberrant activation, you get some cancers, basal cell carcinoma and or medulloblastoma. So I'm not getting into the details. So here you see, so ligands are hedgehog family ligands. So sonic SAH, Indian SSH, desert, they bind to the patched receptor on the cell membrane that inhibits that inhibits SMO, a GPCR-like protein, and GLI transcription factors are phosphorylated and processed into repressors. So target genes remain silent. And when HH is present, HH binding relieves PTCH inhibition on SMO. Activated SMO initiates signaling that prevents GLI cleavage, and full-length GLI activators accumulate and enter the nucleus. And you get the transcription. So this is a pretty interesting case when you when you look at this image. So so you get some sort of proteins that undergo proteolytic cleavage, and your target genes are not activated. But when you get the signal, this signal represses that sort of cleavage, and your protein is able to activate target genes. So one more interesting pathway. NF-kappa B signaling pathway. So you will see that if you study immunology and some sort of inflammatory responses. So I'm not getting into that. You can look these nice pictures. I'll just tell you that different pathways, they may interact. So like detect coincidence. You will see that if you study immunology, there are certain cases when you need two eventual signals that will tell you that something bad is happening, and some part of the protein presented to you is part of some sort of bacteria or something like that, and that tells the cells, I need to take this piece of the protein and I need to process it, well, in an appropriate way, and so on and so forth. So you can get some gated mechanism or feedback mechanism. So these pathways, they may interact. Here are just examples of different signalosomes when you have receptors and some other things collected in one place for better efficiency. So I'm not getting into that. And the final things to show you, well, we have considered different receptors and what happens beyond, and here are just examples of different chemicals that activate these receptors and that are well known to you under the name of hormones. So these are chemicals that work on a large distance, and they activate different interesting things. I'm afraid we are really scarce on time, so probably I'll be not able to get through all of these tables, but anyway, it's not so bad. Just to tell you that there are many different hormones. They are classified certain way. So I have these few tables for you. So you see that they may be synthesized by hypothalamic, and they may be hypothalamic or pituitary hormones, or steroid hormones, thyroid hormones, catecholamines, or eicosanoids. So here they are. Well, more or less some lists of these hormones and what they basically do and what is the clinical relevance. So what happens if you have, for example, deficiency or excess of growth hormone, you get dwarfism or acromegaly, and so on. Here is the pathway of synthesis for steroid hormones. You see, they all start with cholesterol, and I hope you know what is cholesterol, and here is the example of the pathway. How do we get all these hormones? And so they have genomic effects, long duration, and they can diffuse through the membrane. Here are examples of thyroid hormones. Once again, some formulae for you. You see that we need iodine when synthesizing these hormones of T3 and T4. And they regulate basal metabolic rate, growth, and CNS development. So if we have certain problems with iodine, that may cause a lot of troubles, including troubles developing central nervous system. So they are lipophilic hormones with nuclear receptors. So they also may cross all that membranes and something activate or deactivate. Here are some problems that we may have. And catecholamines, once again, they are derived from tyrosine, act rapidly via adrenergic GPCRs, and we have considered that GPCRs as well. Here are eicosanoids, they are derived from arachidonic acid. Here it is. If you have ever heard about COX, that's cyclooxygenase, and probably you know some of the chemicals that inhibit COX and COX-1 and COX-2, and oh my god, I forgot the name of the pills that are often used when you have a headache or high fever. Who can remind me? Ibuprofen, probably. I'm not sure what's the chemical compound. I guess acetyl salicylic acid. I have, yes, aspirin. Yeah, I guess aspirin and acetyl salicylic acid is the main chemical compound of the aspirin, and it is inhibitor of the COX-1 and COX-2 that basically synthesize these sort of molecule eicosanoids from the arachidonic acid. So if you get to the drug bank and read something about aspirin, I guess you will get a little bit more information on that. And the last slide is some sort of different hormones, their primary and secondary targets, and how do they influence everything in the body. So once again, I don't see any reason to get through each of these pathways. You can just look at this table and get the idea. Good. I guess I have even taken a little bit more time than anticipated. But if you have any questions or statements so far, please fire away. I see no questions, no statements. Good. Then thank you for your attention. Have a nice evening and see you next week.